Today I'm going to talk about, should I face that or them? I'm going to look at you. Today I'm going to talk about data, and we all know about data. Data are our best friends. How to get it? Well, we know how to do that. How to keep it? We know how to do that. And how to report it? We know how to do that. So I'm basically here to tell you what not to do. Yes, that is I. But I wanted to thank Brian Weimer and Gabriel Navarre for helping with these slides. Thank you.
It's been like 3 years?
Yeah.
And we were uptown.
Yeah, we were. I'm completely discombobulated.
Yeah, that's good.
Anyway, so, yeah, you can read this, or think about the truth. What is the truth? And we're all scientists here, so let's think about scientific truth. How do I know that transcription is carried out by DNA polymerase? Well, I read it in a textbook. Must be true, but someone had to write the textbook. Where did they get their data? Okay, person writing the textbook probably got it from a paper or 2 or 5 or 20. Okay, but these papers reported it. But how do you know the papers are true? You could visit the research lab of the scientist and talk to the lead scientist on the project, and he'd probably tell you it was true. Or you could press him, and eventually you're going to get back to the lab notebook. Okay, so the laboratory notebook right there is where the crux of everything lies. If you lie in the notebook, the paper, the textbooks, the beliefs, the dogma are all based on fallacy. So there's an enormous amount of importance associated with recording your data and doing it accurately, and then reporting in a, I would say, a non-political way. We just report the truth, okay? We'll let the community decide what it means. We'll hypothesize in our discussion sections in our papers, but it's not for us to say exactly what we showed, just how we interpret our own data. Okay, so today we're going to learn about data acquisition. We are briefly going to cover policies for gathering and keeping data, when you have to report it, when it can remain secret, etc. And then, yeah, we're all going to be interested in looking at these websites. I am absolutely not sure. So, so these are the subjects we're going to cover, okay? How to get the data, how to be objective, your laboratory notebook. My goodness, if I could only talk for 5 minutes today, it would be about the lab notebook. Data errors, and your view on the errors was just spot on. I'm using almost verbatim what you helped with there. Data ownership, you know, when you find something new, who owns it? Do you own it? Does your PI own it? Does Tulane own it? Does everyone who ever helped you own it? We'll cover that briefly, and then finally I'll mention data management. Okay, but first, data acquisition. Okay, so we obtain and record primary experimental information. Yeah, the record keeping, a foundation, subsequent data analysis. It's your notebook. Okay, without good data collection and record keeping, everything else thereafter is tainted. Your discussion with your PI, your discussions with grant agencies, your manuscripts, your papers, your book chapters, everything. So all of the load comes down to us, the people gathering the data. Okay, so preliminary experimentation. Now, there are 2 types of experiments that we do, and I like to divide them up like this, so other labs think of it differently. I have the quick and dirty experiment, and then I have the prime time experiment. So let's say you come into my lab, you're interested in looking at transfection, okay? You already know how to deliver genes into cells, and if you guys don't know what transfection is, I'm a gene therapy guy, okay? It's pretty much all we do in our lab. So we get genes into cells and have the cells express them and alter the behavior somehow because of it. So you come in, and yeah, you've done this before, but you want to try out a new liposome, and you brought it with you. And you've only got like 10 minutes. And you're not even sure this is going to work. You just want to know, is it worth anything? So we could do a quick experiment. We can look at it, and the cells glow green, and we're real happy. And then I say, okay, now we got to do it for real. Okay, you got to have your controls. You got to document every single thing that you do, because what you get from these quick and dirty experiments, none of it's reportable officially. None of it. But they're quick and they're cheap. They're easy. Okay, so sometimes if you just want to try something out just to see, we can get by with that. Okay, confirm, can we get genes into cells? help develop. I was wondering if this liposome versus this polymer is any good. I just want to see them both work once. They're both new. So you come in, the liposome doesn't work, but the polymer works really great. You're going to start to think of experiments focusing on that polymer, most likely. Okay, unless you're a chemist. Then you'd like to fix the problem, right? Interesting. And I guess we're finished.
Let me get my IT guy. There it is. Okay.
Oh, okay. It just wiggled the mouse. Okay. So again, what we get from these experiments is designed to help you and you alone. Okay. But the real data, the formal data, you've got to document everything. You've got to do all your controls, and you've got to do them every time. Okay. So you come in, we're doing more transfection experiments. We always do one where, okay, we deliver the polymer and no gene. And this cell should not glow green. Okay, do we always do that? Yeah, we do. Do we know how it's going to come out? I sure hope we do. But that one time in your whole lifetime, when they start glowing purple and everything else is glowing green, you didn't even really deliver anything. That's the one time you got to watch out for. What did I do wrong? So every control, every time. And then document everything. Okay, why? We get a further understanding of what's going on, the question that we're trying to address, and then, of course, if we're going to have a discussion and conclusion section to our paper, it has to start with documented formal data. Okay, and, this word scares me. Grave moral responsibility, and I tried to change it last night, and I thought, grave kind of sums it up. If you don't accurately report what you see, it can mean the death of your career and your PI's laboratory and maybe several other careers. If you're dealing with a pharmaceutical, it could literally turn out to be a death somewhere. Ask Jesse Kelsinger's parents. Okay, so objectivity. Again, this should be straightforward. We all know to be objective. I'm going to try to cloud the waters a little bit, and then we'll get through the cloud, and hopefully we'll be a little wiser afterward. So what do we recognize as data? We've got qualitative data, we got quantitative data, and we've got potential down here. Now, quantitative is pretty obvious, right? We got numbers involved. We have raw data. You can record it by hand. I looked at the microscope. I saw 153 cells in these areas of my hemocytometer. Okay, that's qualitative. You did it by hand, but quantitative. You did it by hand, but it's still a specific number that you observed. A machine can output it for you. An instrument with NMR peaks, or if we're talking about cell counting, you can put it in the Coulter counter, and it comes up with 154 cells. Aha! But both of those are quantitative versus qualitative. Okay? This might be out of one of my notebooks. The mice in group E were all running around the cage like crazy mice, doing backflips, and it was very entertaining, but probably indicative of some type of stress or stimulation. Okay? I haven't associated any numbers with that, but it's still very important, especially if you're a mouse. You don't want to be treated like those mice in group E were. Okay? Some instruments can give you qualitative data. For instance, you take a picture of some cells, you do histoslides, and you look at them. Yes, we can get quantitative data out of qualitative slides. For instance, we take sections of some material, some tissue, and we stain them for whatever protein we're looking for, and then we can count how many of these slides and what area had what intensity of fluorescence. So you can get quantitative data out of qualitative data. Slides, movies, and then, of course, potential data. So my lab, we test out a cancer treatment, and we harvest the bladders from these mice. We look at bladder cancer. So we can harvest the bladders of these mice, but it's a big deal to do a tissue section, right? So we would want to do all of those one tissue section day. So we'll collect the bladders over days and weeks, and we'll preserve them in OCT, some fixative, okay? And then we'll come back later. So those represent potential data that we can get. Even after we section the blocks, we still keep the blocks, because who knows, 10 years from now, we might have some new ideas, something to stain for. And we can go back to these mice and reuse the bladders. Their treatments are already documented, right? So we can go back and reliably continue to process. So, of course, impartial data, not biased. Of course, that's objective. Not motivated by personal gain, which we'll get to in a minute. And it should rigorously test your hypothesis. And of course, if you don't know, all of your data or all of your experiments should be hypothesis driven if you have any hope of getting funding in the U.S. Let's see. Avoid becoming personally attached to a hypothesis or a concept. Maybe you've got an idea. It's your idea. You came up with it, okay? And you're really rooting for it. And the first few experiments you do support your idea, and you're like, yeah, I did this. And so your PI says, oh, I think you should look at these on such and such machine. We're going to if what you say is true, then we should observe a change in surface charge concentration. And so you learn about a zeta sizer, and you go and you use it, and you're kind of hoping that you're going to get, we'll just say, positive values, because they're going to support what you've been saying all along. Okay, you at night at home can. Sorry, I'm pointing at you. But you can root for that to happen, but when you step into the laboratory, you can't root for anything anymore. Okay? You're just a reporter, and if the data come out all negative, okay, we'll rethink the great hypothesis. Let's see. And you have to accept validity of your report. So if you come to your PI and you say, these really were extremely positive, like I predicted, you're putting your career on the line every time you make a statement like that. But it's okay, because we're all scientists, we're all objective, and we're all very forthright in our data. So it's, even though we risk our career every time we make a scientific statement, none of us, I doubt any of us, I would bet none of us would knowingly mislead people just to prove our hypothesis is correct. We're reporters of science. Okay, so told you we were going to get back to personal gain. You want to make your boss happy. Your boss is happy, you're happier. You're still working 16 hours, but you're happier. Or maybe you are the boss. I'm not assuming you're a student. Maybe you are the boss. And if you get the data, you can get the grant. You get the grant, you get tenure. And then all your problems leave, right? Yeah. Personal recognition. That was a fantastic hypothesis. You're brilliant for having come up with it. We are so lucky to have people like you in the world. It kind of feels nice to hear that. And there is that little temptation for this type of positive reinforcement of negative actions. Improve your chances for grant funding. If only we could prove this one extra thing, it's more likely we get our paper into Nature Biotech. Okay, so you do the test and you get iffy data, but you still report them to support your hypothesis. That's not really the way to go about it, right? You probably get rejected anyway, but that's good. They're checks and balances. So Nature Biotech is going to say, you did kind of iffy data, and they won't accept it. Okay, so don't even waste everyone's time. Report what you see. Yes, one of my favorite quotes from my whole career so far. We had a German postdoc once who was reporting something, and she had experiments that just didn't work. And she was up there explaining everything. She got to the one slide where it didn't really support what she was claiming was going on. And after she presented, she goes, these are the data. We can't change them. And she went on merrily. But it really stuck with me. The data are the data. We are just reporters. Okay, so I've got a few little vignettes, and I think the vignettes are the fun part of this talk. So let's do the first one. It has to do with personal gain. So I'll read it to you. That way you can close your eyes and rest. A principal investigator outlines her theory for a certain effect to her graduate student who's involved in acquiring data to confirm or refute this theory. The PI explains her anticipation of finding experimental data having these values. Graduate student generates the experimental data with approximately the anticipated values. The data are published. The PI gets recognition. Her grant's renewed. The graduate student receives her PhD. All these appeared on the personal gain thing. There's nothing wrong with personal gain. You should be rewarded for good work, right? However, in this situation, the next student who comes in after she graduates comes in and has difficulties reproducing the data. That often happens. You take over a project. It's hard to get the experiments up and running. However, after further investigation, it's found that the first graduate student chose, whenever the data were ambiguous, only chose the values that came closer to the predicted values, thereby overemphasizing a trend that may or may not have existed, that the data that was not present, that the data weren't showing in the first place, okay? She threw out the muddled data, kept the choice peaks that supported what she was saying. Now, we have a problem. Should the PI have restrained herself from mentioning her anticipations? That one's tough. Okay? Now, if I'm talking to a fellow scientist, and he's going to do work as an independent laboratory for me, I might blind him to what I think. Okay? I'll lay out the problem and say, when we're testing this, and he goes off and he looks at it. However, if you're my student and you're a second-year graduate student, and we're looking at such and such, what I would hope my second-year graduate student would say is, why? Why are we looking at this? And I'm going to map out the foundation for the entire thesis project for my graduate student. Okay? Graduate students are smart. You're going to catch on and you're going to think, so you think if we manipulate the expression of this receptor, we're going to get this response. Well, I think it might happen. Let's see what happens, okay? So in terms of a PI-student relationship, I'm not real sure that projecting your expected values for the data was completely wrong because of the pedagogical aspect. But when we go into a lab, and I've said this how many times now, we're just reporters, okay? You should, as a scientist, you should never expect a result. You should never root for a result. Now, I wear also an engineering hat, and it's a little bit different that way, okay? I'm testing an anti-cancer drug. I'm going to hope that all the mice live, okay? But if they die, I'm certainly going to report it. Should the PI have more closely supervised the first student? Should the PI have insisted on more thorough check of data reproducibility? What should the PI do now? That's the hard one. Okay, so yes, the PI must supervise the design of experiments and the processes of acquiring, recording, examining, interpreting, and storing data. Accordingly, the PI must acknowledge negligence on her part. Now, that's easy to say. Any PIs in the room? Okay, I'll tell you as a PI. I don't have a lot of time to go up and watch you do an experiment. If I carve out time to get into the lab, it's so that I can do an experiment. Okay, so it's not realistic to expect your PI to sit there and say, you're holding your pipette at the wrong angle. That's going to alter your data. Okay, a lot of graduate training is about you have to figure out your own way. That's what a PhD is all about. Are most of you PhD students? Nobody is. What kind of student are you? Yes?
I'm a student.
Postdoc? Postdoc. Okay, so you got your PhD. So we know PhDs are all about finding your own way. Okay. Now, scientists have a duty to avoid contaminating the literature with incorrect information. At this point, the PI needs to get involved, okay? It's not that you're going to submit a retraction immediately. You're going to get up and see what's what. You've got grad student 1 who gave you good data, grad student 2 who says, I think she cherry-picked her data. So you're going to get involved, and you're going to find out what's what, and you're going to supervise experiments to clarify, were the data cherry-picked? Were they good data, and the second student just isn't up to speed on the project yet, okay? However, if we do find out the first student cherry-picked, it's on you. You got to come forward and you got to publish a correction or a retraction. And that's really, really tough. I've never been faced with that, but that's why PI should be involved with their students a lot so they know what data they're getting, if they're having trouble, if everything seems a little too perfect, you might want to see the notebook, etc. This is one of my favorites. This is another case study. It's about outliers, okay? So consider a PI acquires data that portray a wonderful correlation between administration of a drug and a physiological response, okay? However, about 10% of the data are really far from the values you predicted. So you predicted a nice bell-shaped curve, dose-response curve, okay? Get beyond the optimal, you get bad effects. And everything's fine. However, over here you've got a sprinkling really positive or negative data values, but they don't fit that nice curve. What do? Well, there are explanations as to why these data are different. For example, some experimental parameters might not have been well controlled, and they were different for the experiments in which the results deviate. The PI chooses to ignore the outlying data for the publication. Actually, I've changed this one. Not necessarily bad move. And I'm glad you said that, because this is a very, very important and subtle topic. So should the PI have published the outlying data with an explanation, thereby weakening the conclusions? It depends on what was different. Okay? So the experimenter may or may not have erred in throwing out the outliers. Now, just because a group of data doesn't fit your hypothesis is not a good reason to throw out data. In fact, your rule of thumb should be never throw out data. There is one very specific exception. Okay? If there's an objective reason that could justify discounting the data that you could have noted before any data were collected, then the group can be removed from statistical analysis. Okay? For example, all the mice in group E, remember, they're running around the cage doing backflips? Okay? It turns out that they had escaped from the cage the night before and ate some reagents in the lab. Okay? Those are disqualified from the study. They've eaten reagents, they've eaten it was originally a tube insulation off of live wires. That was kind of funny, but I was told not to do it. So I said it anyway, now I'm in just as much trouble. Okay, so you've got mice that got out. These mice are disqualified from the study. I'm sorry, okay? You shouldn't even take them to the end of the trial if you can at all help it, because what good are the data going to do for you? Unless you wonder, they ate half a pound of glycine I had laying around, and look, they're all cured. Sometimes you run into discoveries that way. Okay, so the most exciting discoveries come from unexpected results. You're not smarter than nature. Maybe the confounding results are reflecting something you haven't thought of yet. Okay? The healthiest and safest and most exciting approach to take to outliers is something happened I didn't think of. Maybe no one else has thought of it either. Okay, the notebook. We all know what notebooks are. We've used them for years. Part of this series, this part has to be said. Okay, so you can continue to sleep. You're just not having a good time today. Yeah. Yeah. There we go. Trust me, it's like this in all my classes. Okay, so the library notebook is the primary record that you set down. It occurs as it occurs in the notebook. Okay, I don't even need to read this. What's a good notebook? Well, you can't add and subtract pages. So loose leaf, free ring binders? No, it's got to be okay. Once you have the bound notebook, you can't go ripping pages out because that looks suspect. Okay, every page must be numbered. If you want to save money and get the 99-cent composition book, well, in my lab that's acceptable. Okay. The problem with that is it's not acid-free paper, so after about 15 years, the ink's starting to turn funky colors, which is entertaining, but maybe the grad students don't like that. So number the pages, even if you have to do it yourself. Date your entries. When did something happen? For one reason, you go back and you're like, it was around Christmas. I know I got these data and you can look them up that way. Or patent issues. Okay, you might need to support when a discovery happened with original notebooks that have been dated. You write them in ink, not pencil. Okay, why? Because pencil's easy to erase. So no erasures. No obliterations either. If you make a mistake, you write 83 instead of 38, and you know that's wrong. Don't go 38. Just put a line through the 83 and write 38 next to it. Maybe even initial it if you want, if you're really scared. There has been a case of a student, I think it was a postdoc, but it might have been a grad student, who obtained a science article, okay, was an author, first author on a science article, and the data couldn't be replicated, so a challenge went up to the data. And the investigators went all the way back to the laboratory notebook, and legend has it, carbon dating was done on the ink, and found that the ink was very fresh compared to the ink in the rest of the notebook. I don't know how sensitive carbon dating is, but that's the scare story they told us in grad school.
Well, and it's so the Office of Research Integrity, that is the office that looks into research misconduct allegations for federally sponsored research.
Mm-hmm.
And so having processed research misconduct allegations, they want to see the lab notebooks. I mean, even though the panel here will look at them, eventually when we send everything off, they look at them. And I know that they test them.
Okay.
And so, yes, it's not just a scare tactic.
Okay.
And
I don't know about the carbon dating.
Well, they do a lot of different stuff. But all of those things on the list are things that we have seen.
Absolutely.
In conducting a research misconduct investigation. So again, research misconduct is fabricating data or falsifying data. So if anyone ever accuses you of that, you want to make sure you don't have a problem with your lab notebooks, because you need to be able to go back and show when the experiment was run and not have missing pages or no dates.
Or big scribbles.
Nothing's dated, so then you can't,
The date's easy enough. The scribbles are my big issue.
Yeah.
Okay. Put a line through, because 83 would have hurt your hypothesis, but a 38 conveniently is exactly the mean. And you say, it's dyslexic. I flipped the number. See? 83, 38. And it does help cover you. Okay? So pretend it's a full moon and he sprouted fangs and he's looking over your shoulder. Okay? So just make everything obvious that you're doing your notebook. Can't wait to see you in about 3 weeks.
All right.
Let's see. Nick. Inserts. You may have inserts. I'm sure you all know that, especially since you're probably all postdocs because no one's pursuing a PhD. But you put in an insert. Let's say you take a picture of a gel. Okay? And yes, you glue it in your notebook. Sign the insert so that half of your signature's on that insert and half of it's off, so that someone can't say, well, you just took it out and got another prettier gel and put it in after the fact. It's just covering yourself. No pages turned out. So that's the scary part about notebooks, but probably the number one reason we have notebooks is down here at the bottom. It's so that someone else coming in can repeat your experiments. Okay? People ask to see my notebooks all the time, even back from grad school. I graduated in 2000, and they still ask to see my lab notebooks. And I'm fine with it because, one, I teach them how, number your pages, date, But second of all, this is what we did, and this is the result that we got, okay? We're using different genes, different excision techniques now, but the ideas are the same. So I'm free to share my old notebooks. My current one, maybe with a colleague, but not a competitor.
Who will ask you to see the old notebooks?
My students.
Okay.
Yeah. So we don't have a postdoc in our lab. So I'm the PI, but I'm also the postdoc. So the students will want to do a technique. I don't have a lot of time to go up, so I'll bring into the office. I'll say, look at these numbers. I want you to figure out how I achieved them so that you can achieve your own. Go to the experiment, bring it back to me, and we'll talk about what you got. Okay. Be clear for your own credibility's sake. And this is in more than just laboratory notebooks. So thesis proposals, grant applications, postdoc funding applications, all of that's important. This is evidence that you thought of it beforehand. Okay, so you go and you get data and maybe there's a patent issue. Some other lab comes out and says, you stole your idea from us. And you can produce your notebook that shows, look, 5 years ago I was working on this. I didn't steal anything from you. Okay. So these should also be clear. Your raw data, strip recordings, computer printouts, assays, blots, your conclusions. I write conclusions in my lab notebook. Okay. Not like we did in freshman chemistry. Okay. Those were a little overboard. But in my own notebooks, every once in a while I'll have, this is what I know so far. I had this, I get this result. I added that, I got that result. But when I add them both at the same time, I get the first result again. This is what I think might be going on, okay? So that later on, someone's trying to figure out what you were doing, maybe the postdoc that follows you. They say, here's the clear line of reasoning. Here's the hypothesis, the new hypothesis based on these three observations, which I now know is incorrect, okay? But I see where she was going. Okay, case study 3, the notebook. Smith, a chemistry graduate student, begins a laboratory research project. At the start, Smith discusses the project thoroughly with his thesis advisor, who also provides relevant references for the student to go read. Go read these papers. The advisor, however, does not mention any lab notebook practices. Okay, so you come into my lab, we talk about the project, I give you papers, say, okay, you're all set up, go, work. And you go and you work, okay, and you soon get interesting results. You come in, we talk about the results regularly, and you keep getting these results, and I'm getting pretty excited about them. You are too. After about 6 months, you get to hear those words that we all want to hear. Write it up. Those are good words. Okay, so you write them up, and the publication will then be expanded for maybe part of your thesis, even though you have your PhD. In Smith's draft, the raw data had been processed into graphs, tables, and text. Okay. Now, when studying the draft, the advisor has a number of questions about the raw data and asks to see the notebook. To his dismay, that's a good word, the advisor finds that the student didn't keep a notebook. Smith just kept records on loose pieces of paper or laboratory paper towels. It's a habit that all of us in our lab do. What few records we do have are undated, and many of them cannot be found because they're on loose pieces of paper. Okay, even if they could be found, I don't know. Actually, I do. We don't know. What are we going to do? And who's responsible for this conundrum? First of all, I'll tell you without even turning to us, all those data qualify as a dirty experiment at that point. Okay, you can't use them. The processed results unsupported by your original raw data are unpublishable. Now, you haven't completely wasted your time. You have the knowledge. You just can't prove it to the rest of the world. Okay, so you've got to go back and you've got to repeat the experiments. Now, maybe you don't repeat trials A through D because you know they never worked in the first place. Okay, and you've got to get the good data out. So you do your control when you do E and a couple others that were interesting. Okay, so you do get the benefit of prior knowledge, but if you're going to publish these, you got to get your original data in a credible source. It should be the lab notebook. Okay, can't even use it in your thesis. Now, whose fault is it? Well, experience shows this actually happens quite often. I told you in my lab, people write stuff down. In fact, I write stuff down on these papers. Okay, am I bad? Maybe not as disciplined as I should be. My notebook's in my office, but I'll tell you, I do this. After I jot down everything that happened in the experiment, I take that paper immediately down to my office and transcribe it into my notebook. Okay, I don't leave the office until it's in there, because if I do, if I forget one detail, it invalidates the whole experiment. Okay, so I copy everything down. The loose papers those paper towels are not the way to go. Let's see, what else we got? While it may seem reasonable for the advisor to assume that Smith had learned about lab practices as an undergraduate or from his peers, you're a postdoc, I would assume you know a notebook. And she nods yes, and who am I to doubt her? The advisor can't be excused. I have to know. It's my lab. If it's data that are going to come out of my lab, I have to know. So you should look at your notebook once or twice, right? Let's see. At the outset of the project, it was the advisor's responsibility to make sure that Smith had a notebook and knew how to use it. But even though all of the brunt comes down on Johnson, well, the advisor's now named Johnson. Even though the advisor takes the brunt of the problem, he's got to pay for the experiments, Smith has to do the experiments. So Smith isn't going to graduate as quickly as he was because he's got to go redo things. Okay. However, I'll contend your error bars are going to be smaller because you've done it all before and you know the little tricks that will take the variance out of your experiment. Don't feed cells in the morning and then come in and feed them at night instead and expect the same result. There's a little hint for you. It's free. It cost my grad students 5 years to find that out. Okay, let's talk about scientific errors. Okay, different levels of error, all of them we've heard of. I do want to point out a couple in particular, especially skew. Okay, skew is where your experimental design favors results that will yield such and such answer. For instance, if you use Google to search for your references will be skewed. Okay, because Google is not an unbiased search. There's advertising involved with Google. Certain words mean more than other words in the Google algorithm. Okay, go to PubMed. Okay, you get 10,000 hits. Good for you. Okay, read them all. Yeah. Okay, but you want to make sure that you do not have experiments set up that are going to skew your results. Okay, at the experimental level, undesirable and negative results, do not disregard them. Okay, did you do something wrong, or did the mouse get out and eat the glycine overnight? Okay, mouse didn't get out and eat glycine. You might be onto something interesting. So we've already talked about that. At the analysis level, wrong statistical test. As a reviewer for papers, okay, one of the most irksome things to me is someone has 5 groups and they're proving significance between the groups with the t-test. Okay? What's wrong with that? It's for pairwise comparisons, and so you compare 1 and 2 and 1 and 3 and 1 and 4 and 1 and 5 and 2 and 3 and 2 and 4 and 2 and 5 and 3 and 4 and 3 and 5 and 4 and 5. Keep in mind, the t-test, you're saying your results have a 95% chance of being certain, right? Which means a 5% chance of error. And another 5, and another 5, and another 5, and another 5. So your confidence, I'm getting a little math here, if you have a 90% confidence interval and you do 2 t-tests on the same group, you have a 0.95 times 0.95 chance of being certain, right? Which is less than 95%. Okay, you can do that for 2, 3. Don't do it for 10. Use an appropriate test. An ANOVA test would be appropriate for groups. Okay? If you don't know which test is appropriate, find someone that's aware of it and learn, because we're also all here to learn. Oh, it's very irksome, let me tell you. Okay, what else do we have? Grouping is forced. The interpretation level, your personal bias, we already talked about that. I work for a company. My company has $10 million invested in developing this drug. I want the drug to work. Am I going to look at the data and try different statistical analyses until I find the one that says what I want it to say so that I can get more investors and glean more profit for the drug? No, not the way to go. And then finally, fraud. This one is death to a career, death to a lab. None of us in here would ever do it. Just to be accused of fraud can hurt someone's career. Okay? So it's a difficult allegation to make, a tougher one to have levied at you, and even worse if it's proved true. There's a bounty out. When the NIH has a $3 million bounty on a scientist, do you know about this? Guy was caught falsifying data. The grant was supported by NIH. The NIH asked for his money back. The person fled to India, which doesn't have reciprocity with the U.S. So the NIH has put out sort of a warrant or a bounty on this guy. If he comes back to the U.S., he's to be apprehended and billed for $3 million. Fraud's bad. Don't do fraud.
Fraud allegations is why you want to make sure you follow all of those rules on the lab notebooks.
And I'll tell you, I've never seen fraud. I've seen one accusation of a National Academy member. The allegation was dealt with very quickly, very upfront, but for about 6 months, it was the scuttlebutt. He's been accused. Okay, proven to be unfounded claims, but the accuser had unfounded claims. Let's see, other errors. Sampling error. Okay, we're going to use 3 mice and say that we know that our drug cures cancer. No, but there's a reason we do a power analysis. Okay, you have to have enough samples to be able to make a statement. And then selection bias. This one's really good. I like the example. So, distortion resulting from how and which subjects were selected. If you take a poll in front of the National Democrat headquarters, you're probably going to get different polling results than if you go down to the mall. Okay, because one would assume the people coming in and out of the national headquarters for the Democrats would have Democrat point of view. Okay, information bias. Your machine's not calibrated. It might give you results that are consistently too high. Okay, that's a type of bias that isn't personal to you, but it's still bias. Okay, so make sure your equipment's dialed in. Confounding error. So influence of uncontrolled variables that are linked with the independent and dependent variables under study. So, for instance, the incubator ran dry, the cells behave differently. And yes, they do. The mice were affected by the researcher turning on the lights in the middle of the night for another experiment. In fact, that came up in the uptown vivarium. Okay, I had an experiment that was going on in the middle of the night. Someone else was worried, was interested in circadian effects. Obviously, we couldn't turn on the light in the room, so we dealt with it by moving our mice to another room. Okay, but that's confounding error. Tough to find, okay? And sometimes confounding error is silly, or silly sounding until you prove that it's true. We had a joke in grad school, it's the phase of the moon. Meaning there's some kind of confounding error that we just haven't identified yet, but we know it's in there, and then as scientists, we isolate each individual variable and see which one could be causing the result. Okay, and then fraud. Now, there are many different reasons why erroneous data may result, or incorrect interpretations may be made. Mistakes of this nature can be costly, and they have serious consequences, but they're not fraud. That's how serious fraud is. You didn't calibrate your machine. You got consistently high values. You believed that your drug was having this wonderful effect. You published the results. Oh no, okay? You're gonna have to do a retraction or correction, but you didn't commit fraud. Fraud has the element of intent in it, okay? And again, I doubt any of us would ever do it, but it's that serious. Saying you ran experiments that you did not, saying you ran more experiments than you did. Oh, instead of n equals 3, n equals 18. Get your denominator a lot larger, get your error a lot smaller. If your error is smaller, then these bars that were close to each other are now significantly different from each other. You make the claim, you get the paper published, you get tenure, promotions, grants, everything based upon erroneous data that you said you did more experiments than you did. It's fudging on purpose, and it's fraud. Don't do it. Changing the data to fit your bias. Don't get me started. I'm already started. Intentional misinterpretation of the data. Okay, I got these numbers, and of the mice that lived, the ones that received the treatment had much smaller tumors than the ones that didn't receive the treatment. I'll just conveniently leave out that 80% of the mice treated died. Okay, that's fraud. It can sneak up on you. Pretend you're talking to your mom or your dad, and you're telling them what you did, and they understand it all. Okay? Would you try to hide it from them? If you would, then something's wrong there. And then finally, data ownership. Yes, sir.
Issue about changing the data. So I would say that like manipulating images and splicing images in Photoshop, which is a lot of what you see when there's a research misconduct allegation, that would fall into that category of changing the data. So,
that's a good point.
Yeah.
I hadn't even considered that one. I'm such a numbers guy.
Well,
but yeah, we do get
a lot. I'd say the majority of the allegations are either plagiarism, or it's manipulation of the data, and often it's with images and via Photoshop. So,
Photoshop's your friend, but it's also your enemy.
Yeah.
So pretty much, in fact, there's a way to track. You should, okay. Our lab, we basically do 3 commands when we get something off the microscope. We do fluorescence. So we get something off the microscope. Was it Control-Shift-L, Control-Shift-Alt-L, and then Control-Shift-B, and that's it. Auto brightness, auto contrast, and auto sharpen, okay? But we also keep a copy of the untouched image, just in case. So if some reviewer comes and says, this doesn't look anything like what we get. Your colors are too sharp. It's obviously this has been enhanced. You can show the reviewer the original slide and say, this is what I did. Bing, bing, bing. Okay, that will help you. It takes a lot more memory, okay, but memory's cheap these days. It's not that big an issue anymore. And then finally, data ownership. This one can go really, really quickly if we want to, because there's basically one answer. Tulane owns everything. But I do want you to know a couple of historical things. So we've got the Bayh-Dole Act. Okay, it used to be you have federal funding, government paid for these data, the government owned the data. It makes sense. Okay, The problem was the government wasn't doing anything with the data. So it was really hard to, say, start a spin-off company off of data that arose out of NIH-funded projects. Okay? The Bayh-Dole Act says, okay, the data, yeah, you're kind of beholden to the government, but we're going to give the intellectual property rights to the institution that managed the money for you. Okay? And then this is just to be complete, this very long OMB Circular A110. It regulates the use, ownership, and sharing of data and other intellectual property, okay, that were funded by the federal government. It includes the rights of the public to get access to your data. And that's the big point here, okay? The public does have some recourse through the Freedom of Information Act. So what's to prevent your competitor from saying, Freedom of Information Act, show me your notebook? Well, there are, and I'm going to get into what you can keep secret and for how long. Okay, but the Bayh-Dole Act, I told you it's so that we could get these technologies out into the marketplace faster. It removed obstacles that previously blocked the use of technologies. Before this, here it is, fewer than 5% of the 28,000 patents held by the government were licensed, less than 5%. However, I don't know the figure, but once these data were given to the institutions where they originally obtained, I think Tulane's got a very active Office of Intellectual Property. And they will file often. They're very happy to file patents, okay? So I don't know the actual number, but it is far higher than 5%. Okay? Now, usually universities retain it, and they're free to market it. Here are the ones that I told you were going to just love seeing. These are the links to websites where you can get more information about, well, here's the Tulane Research Misconduct Policy, the IACUC office, okay. Let's see, what's this one? Procedures for its Human Research Protection Program. You guys, I'm talking to you, not you, Abhinav. You guys often work with human subjects. You may never see the human subjects, but you'll get data or questionnaires or something from human sources. Okay, the identity of the humans is fiercely protected, and rightly so. Okay, so if you receive one of these requests for under the Freedom of Information Act, okay, talk to him. Definitely go to the brass when that happens. You're talking about laws, and I'm not a lawyer, I'm a scientist, so let's let the people who are used to these issues deal with it, okay? You might have to provide some data to the requester. However, some of the information you do get to keep secret, so you don't have to worry about me coming into your lab demanding you see your notebook under the Freedom of Information Act and then walking away and out-competing you, okay? And the rules are complex. They change all the time. Again, we're scientists. We're not lawyers. That's why we have an office that takes care of it for us. Okay? So who owns the research? Tulane. The idea is a research dollar spent, what does it buy? It buys knowledge. It buys data. Okay? Whoever provided the money owns the data. Now, it's the government. We just talked about the Bayh-Dole Act, so it's Tulane that owns it. Okay. Sometimes you'll have a company, like maybe Toyota comes in and they're interested. I'm downtown. This is useless. Hey, Abhinav, maybe Toyota comes down. He's gone. No, there he is. Maybe Toyota comes in and funds a project for a polymer that you're working on. Okay. The data are owned by Toyota. Most likely, Toyota and Tulane will work out an agreement up front before you get any numbers. Okay, so there's no confusion there. You guys, you don't have to worry about that, usually. Yeah, who manages the data? Well, of course, whoever manages the lab is responsible for it. You have the lab manager, it's the PI. Okay, it's the PI's lab. PI is responsible for everything that goes on in the lab. You got to keep your data for, well, I'll tell you now, for at least 3 years. That's ridiculous. Some people say, oh, well, it's supposed to be 3, but save it for 5 just to be complete. I never throw data away. I've got my first grad school notebook, okay? I'm never going to use the data, but I still have it, and I see no reason to throw out data ever. So, in the remaining minutes, let's look at this technology ownership one, because I have undergrads come to me all the time. I teach a biotechnology course, and we cover patents and licenses, and sometimes they're infuriated because they want to get their $10 million. Well, let's read this one and see what we see. So, a graduate research assistant works on a sponsored project that financially supports 50% of her time while she pursues a PhD. Okay, she's federally funded is what we're going to say. Her advisor, the PI, developed the original idea and has predicted some correlations. And you got an undergraduate helping you. Now, research turns out to be scientifically successful. You get a new process out of it that promises commercial value through patenting and licensing. The graduate student finishes her PhD and gets a copyright for her thesis, for which the relevant correlations are reported. Now, the university decides to file for a patent application. The graduate student says, I came up with this idea, and I have a copyrighted document with my data. I published first, okay? So she wants to file for a patent application herself because she's found a company that wants to exploit the idea. The graduate student argues that she came up with the idea, she's got the copyright, she published the copyright first, no patent's been filed for, therefore she owns it. So, is the graduate student entitled to pursue the commercial interests from her thesis research? What do you think? Okay, no. Because, and this is the official word, because I'm about to give you an exception, okay? But the official word is the graduate student is considered an employee of the university, okay? If you worked for Toyota and you found something out, you're not going to file for it. Toyota owns your discovery. If you work for Tulane and you come up with a discovery, Tulane owns your discovery, okay? Can the PI run off and file a patent on his own? It was his grant. Well, with an exception there, too. So let's say the PI and Tulane choose to file for a patent, okay? And they file the preliminary patent, and he has a year to come up with supporting data while they do, searches, patent searches, to predict what kind of arguments the patent reviewer is going to come up with. And after almost a year, Tulane comes in and says, actually, I think that we're not going to be able to beat such and such claim for originality. Okay, so we are giving you the right to go forward with the patent. Okay, and if you choose to go forward with the patent, yeah, you pay the costs from then on, but it's your patent. But Tulane will be very clear if that happens. Okay, but I'll tell you from experience, you would rather have Tulane on your side. And if they think that you don't have something that's going to beat, counterclaims, they have more people, including lawyers, working on this. You probably better take their advice, okay, and just keep creating. As scientists, we're not in it to create one great idea that's going to change the world. Our product is our ideas, and we should be putting out product all the time. So don't get married to one idea. Keep putting them out. We're all creative in this room. Okay, so the grad student entitled. No, of course not. Neither's the PI. What about the undergrad? Let's say a patent's filed. Let's say the university and the PI's real cool. And he says, you're right, former grad student who now has a PhD. I want you on the patent as an inventor, okay? And the grad student says, what about me? I was the one that came in between basketball games and did an assay once. Usually the bar, you have to add something intellectually novel that is being patented, okay? The student may say, but no, I decided if we use the second rack in the incubator instead of the top one, the cells are going to give more reproducible results, and that's what we did, and we got the results, and therefore I gave something intellectually novel to this. Well, we're not patenting putting cells on the middle versus the top shelf of the incubator. We're patenting this drug that we designed, and you can't even draw the molecular formula up, by the way. We're patenting this, okay? We appreciate your work, and you're certainly part of the paper, either as co-author or acknowledgement, one or the other, but yeah, patents get real tricky. If you have an undergrad that gets real upset, I guess I'm talking to the uptown people again, who look like they're about to leave. Just know you don't get rich off your patents, okay? The patents are a mark, just like a paper. You publish a paper, it's proof that you've done something interesting that the community can value. A patent is much the same thing. You're not going to get rich off your patents in general. Okay, so let's go through this. The discussion. Inventorship is reserved to describe the person, people who have created something new. We just talked about that. Ownership refers specifically to the person, company, or institution that holds title to the patent. Okay? It's just trace the money back is basically what it says, keeping in mind the Bayh-Dole Act. Okay, so even though the money may have come from the NIH, it will be the institution that's managing all of that. Okay, thus, even if this idea was developed on the graduate research assistant's own time, and the PI and the undergraduate research assistant did not make an intellectual contribution, the university still has title. And it all goes back to who was the employee. Here's the exception I promised you. An undergraduate is not an employee of the university unless the university pays the undergrad. Okay? Let me split this hair for you. Undergrad in your class comes up with an idea, comes up to you after class and says, hey, I have this idea. I'm going to start pouring motor oil on my tomatoes and then get biodiesel tomatoes that we can burn directly on a stovetop. You ever heard of that? No. Student goes off, pursues it, gets data for it, and decides to patent it. It's the student's. It really is. However, if student does the research in a Tulane laboratory with Tulane money, then Tulane's got claim. Okay? Then it gets sticky. But that's the one counterexample that I can come up with. Okay, and data management, keep your notebooks. Thank you for your attention, and have fun with the rest of the semester.
Any
questions? Primate Center's gone. Avanah's still there. I see him. He's just right here. Okay, thank you for attending.
All right, thank you.
Sure, it was fun.